Hello, so we are going to continue the replication process. In the last video we talked about DNA replication, the 5' and 3' and the structure of the DNA molecule in details. So now we are going to talk about the replication process. So in DNA replication, one DNA molecule is basically producing two double helix DNA molecules, one original strand and a new strand is forming, as you see in the picture right here. Okay, so this original DNA molecule is the parental strand, and this has one parental and one daughter strand, and that's exactly the same identical DNA molecule. So let's start talking about the replication, what it requires. So what basically it requires is number one, free nucleotides. Okay, these are like the raw material if you're trying to understand. And then we need the parental strand. Okay, and then it requires various enzymes. Okay, and Rna Primer. So these are the basic stuff that we need for DNA replication. Okay, these are the things we need. So now let's talk about the steps. Okay, so the steps, there's three major steps. First is the initiation, and then elongation, and the third one is termination. Also there is another step which is normally not mentioned, is proofreading. And we'll talk a little bit more in details about all these steps. So just to get an idea what we're going to talk. Okay, so let's focus on the enzymes. There are various enzymes we need for this process to start and also to make new daughter strands. Okay, so let's start with the first one, is the helicase. And there are various other enzymes also, but we're just focusing on the major ones. Topo Isomerase, also called gyrase, and then DNA polymerase. We're just going to write poly. Okay, let's write it polymerase 3. Then SSB protein, which is single strand binding protein. Then RN, this is the short form, ribonuclease H. DNA polymerase. I'll write it in short, polymer 1, number 1, okay. Primase and ligase. I think I'm not forgetting anything else, the major ones. Okay, so these are the enzymes. Okay, they take active part in the DNA replication process. Enzymes, you know, they are biological molecules, acts as a catalyst. Okay, they speed up the reaction. So now um, let's focus on the steps, and then we can go through side by side with the function of the enzymes. So the initiation step, okay. So the initiation step, let me change the color of my so for the initiation step, I'm sorry, it starts with in the DNA double strand as, let's call it, origin of replication. So the replication starts right here, like in one of, you know, one of the spot, let's say, in the whole strand. So origin of replication. So that's the first stop. I mean, our first spot. That's where the replication process starts. Okay, and this is started through some initiator proteins. Okay, initiator proteins. There's a bubble formation, and there's a fork formation, and then, you know, this starts the process. So the first enzyme, that's the helicase. We mentioned the enzyme, the helicase. It, say if this is the strand. Okay. So, and you know, there's hydrogen bonds here. So the helicase enzyme, it breaks those hydrogen bonds. The hydrogen bonds are not very strong bonds, and so the helicase comes over here and breaks this helicase. It breaks those bonds. Okay, breaks the hydrogen bonds, and once it's broken, there is a enzyme, Topo Isomerase, the second one in the list. What it does, it untangles the strands, so it doesn't get tangled, because DNA polymerase needs room to add the nucleotides. Okay, and then the our elongation process starts. So let's say now the DNA molecule is like Y-shaped, like this. Okay, so there's two strands. This is parental strand, let's put it PS1, and PS2, parental strand 2. Okay, and this are the nucleotides, I mean the base pairs. So say if this is A, this is G, this is C, like that, and this was T, and G is C, G, like that. Okay, and similarly there are other base pairs right here. So it's separated. Now the SSB proteins, there's a single strand binding protein, they kind of anchor the SSB, that holds them apart, and also lets the DNA polymerase 3 to come over and start acting. So on the nucleotide, so they can start putting those nucleotides. But before that, there's a primase, which is a protein. The primase comes and it lays down the RNA primer. Okay, DNA polymerase 3, it cannot really start its job because it doesn't have anything to attach itself to, or you can say that it needs base pair to start the initiation process. Okay, so it lays down the RNA primer. So this RNA primer, let's magnify it a little bit. So this is the 3' end and this is the 5, and this is the 5' and this is the 3. We already talked about this 3 and 5' ends in the previous video. If you haven't watched, you can go and understand, just listen to the video, so you will understand what is the 3 and 5' end. Okay, so the RNA primer, and it starts, say it's here. Let me change the color. So this is the RNA primer right here. Okay, that's the RNA primer. So there's another thing, is when the helicase breaks down those DNA, I mean the strands, the hydrogen bonds between the nitrogenous bases, say this helicase is going in this direction. Okay, so the RNA primer is laid down right here at the 3' end. Okay, the hydroxyl, the free end. Okay, so the reason is this DNA polymerase 3, it can be added as, let's see, be added as 5 to 3. Okay, so it's sometimes very confusing. If you look at here, you might say that this is 3 and this is 5, so how you're saying 5 to 3? 5 to 3 should be opposite way. No, actually the thing is, we are making a new strand. Okay, there's a daughter strand. The daughter strand is going in 5 to 3 direction. Okay, so this 3 and 5 is the parental strand, like we mentioned up right here. Okay, DNA, these nucleotides, it is read, like they read it, from 3 to 5. Okay, so read 3 to 5, but adding is 5 to 3. So this is 3 and this is 5. When you add another strand here, so it has to be 5, because DNA in the structure you have seen previously that there's one, or let's look in the picture over here, is this is 5 prime, this is 3 prime, 5 3, 5 and 3, and in the bottom is 3 and this is 5. Same way here also. Okay, so if we are talking about this, we are talking about this strand down there. Okay, so that's the original strand or the parental strand, and we are making a new strand. So when we are making it, then it has to be like 5 on the top, like this one. It cannot be 3. So that's why it's added as 5 to 3, direction, and that's why I name it, and similarly here it is going to be added in the opposite direction. 3 to, it's still 5 to 3. Okay, I'm sorry. Okay, I'll explain it here. That might be confusing. So this is the 5 end and this is the 3 end. Okay, so it will go in this direction. So I should have written 5 3 in this way. Let's ignore that one. Okay, so it's 5 and there it's 3. So it always runs, it always adds the nucleotides in 5 to 3. So that's the most important thing. If you understand that, then it's very clear that there is no confusion between the leading and lagging strand. So this is the most important thing to remember. So remember this. Okay, so it's always 5 to 3. So here it's 5 to 3 this direction, and here it is 5 to 3 because it's running in the opposite direction. They're anti-parallel, so that's why the addition is also in a different direction. So that's so we talked about helicase, Topo Isomerase, DNA polymerase 3, SSB protein, and we talked about the primase. So this RNA primer, let's talk a little bit more about it. It's a tRNA. It's a transfer RNA. Okay, so it lays down, like it's laying a brick first to start the building, something like that, and then from here DNA polymerase 3 it picks up. Okay, and this, since the direction of the helicase is from left to right, okay, and the RNA polymer is right here, so the nucleotides are added continuously. So it just needs one RNA primer. So RNA primer, it just needs one on the top strand, and then it goes continuously because it's following the helicase, the enzyme, which is going from left to right. So it's in the same direction. The chain is opening, I mean the zip, opening the DNA. But down here, it's in the opposite direction. So let's focus on that. The purpose of the video was this leading and lagging strand. So let's draw here. Let's try to understand. So this is the 3, this is the 5, this is the 5, and this is the 3. Okay, and the helicase is going in this direction, and here is the RNA primer, and after that the DNA polymerase 3, it keeps adding and making a new strand. Okay, but here this in this 5 end, and this is the 3 end, and this this DNA polymerase 3, it only adds 5 to 3 direction. Okay, so in this, let's focus this way. So this would be added. So it's, here is the RNA polymer, the first one, primer. Okay, so this is the RNA primer. So we are going to start the addition in this direction because it has to be 5 to 3. So when it is added, then DNA polymerase comes and it adds the nucleotides, and there's here, and just imagine that this is just a bubble here, and it's opening. Just focus on that. This is opening in this direction. Okay, so let's say in this picture, this is picture number 1, and this is open, so only this much. Let's say it has opened, say 5 centimeters, just something to understand, at 0.001 second. Okay, now let's draw another diagram in the bottom, and okay, so these are the nitrogen's bases, and the strand is going smoothly all the way. Okay, and this is the 3 end, and this is the 5. This is 5, and this is 3. Now these are the nitrogen's bases. So now here we had added, say, right in this area, first RNA primer. Okay, and after that polymerase DNA 3, it added a segment right here, and here it is closed, you can say, or it is there. Okay, so this is, say, after 1 second. Okay, now initially it was 5 centimeters. Let's say this is 10 centimeters. The chain has opened. So since the helicase is going in this direction, there's a lag always here, because it's adding and it's opening in up, but it is adding in the opposite direction, because this is 5 and this is 3, 5 to 3 prime of the daughter strand or the new strand. Okay, so this is going in this direction, so there's always a lag. So when after the time elapsed, say 1 second or 0.1 second, then this is again, there's lagging here, so we have to add another primer, RNA primer, and then DNA polymerase comes. So these are short segments, and there is delaying, is going slowly as compared to the top one here. So it requires multiple RNA primers, this strand. Okay, so it requires multiple RNA primers to copy the strand. Okay, so, and then say this elongation, and then eventually it ends reaches the end at the telomere, and then replication process stops. Okay, so now it has caused some gaps. Okay, because of the so many RNA primer, they are just temporary, hybrid that was added, DNA RNA hybrid that was added there, so to start the process of the replication, adding new nucleotides. So these are the gaps. Okay, these are the gaps because eventually these RNA primers, these are hydrolyzed first by ribonuclease RNase H. Okay, so these are hydrolyzed by RNase H, another enzyme. Okay, and once it's hydrolyzed, they are removed by DNA polymer 1. Okay, and that causes the gap. It's not continuous strand, and this forgot to mention, these segments, short segments, non-continuous segments, are called Okazaki fragments. They are like Okazaki fragments. Okay, so Okazaki fragments are non-continuous fragments, and the short fragments. So between two Okazaki fragments there is a gap, because those RNA primers, they are removed by DNA polymer 1, because they don't belong there. Okay, so, and they are eventually replaced by DNA polymerase new nucleotides right here. But still there is a little bit, gap there. Those are brought together, or glued together, or you can say they are sealed by ligase. Is another enzyme. Okay, so we kind of covered all the enzymes in short, like what it does and all. But the focus was again, which was the leading and lagging strand. So that was the main reason I wanted to do this video, because it could be very confusing sometimes to understand. So this one, coming back to this picture, so this is the leading strand, because it is leading, because it's going ahead and faster, that's why it's leading. And this strand, which is slower and lagging, so anything that lags behind, it's slow, it's going slowly. Okay, so that is the slower segment, or you can say slower strand. But eventually, they become a full strand, and then there are two DNA molecules that that are produced from a single one in the cell cycle synthesis phase, so they're ready for mitosis. I hope I made it clear. Just want to point out another thing before I close this video, is normally you would see in the books and everywhere this DNA structure 5 and 3, 3 and 5. It's in vertical alignment. Okay, but when we see the replication, it's in the horizontal alignment. So sometimes it is difficult to understand where, or it's confusing which one is 5 and 3. So when this vertical we lay it down into horizontal position, so this top one is 3 prime and the bottom one is 5. Okay, and I have shown the replication, the helicase going towards the right, from left to right, but it can go either way, it can go the other way, and then this lagging and leading strand would be opposite. The most important thing to understand and to remember is the DNA polymerase. It adds, or it is added as, it adds the new strand. New means the daughter strand, 5 to 3 direction. So when you look at 5 and 3, that's the new strand, 5 to 3. It's not following the direction of the parent strand. Then it is very confusing. But it is read. It has to be read first when the strands are open. Somebody has to read it, like, okay, what are the nucleotides or the base pairs? Whether it's A, then the T has to come. So it is read from 3 to 5 direction. But when you're placing those, matching those nitrogen's bases and adding it, it is always going 5 prime to 3 prime direction. Okay, so I hope this video helps, and thank you for watching.